Hello everyone, good afternoon. Welcome to the seminar in the UCD School of Agriculture and Food Science. It's great to see so many participants online. So the title of this afternoon's seminar is Digging Deeper, Soils and Greenhouse Gas Emissions, and we have two great speakers lined up for you today. So just a quick reminder, please could audience members add their questions in the Q&A, the chat function, the Q&A function. And at the end, we've got plenty of time for a good Q&A session. So please send your questions through and maybe indicate if it's for Paul, Hannah, or both. So we have two speakers today. So first up, we have Dr. Hannah Cooper. So Hannah is a soil biophysicist interested in linking greenhouse gas emissions to soil structure and soil root interactions. The focus of her research is assessing and mitigating the negative impacts of agriculture on soils and ecosystem functioning, and proposing and delivering viable environmental solutions. Her research spans a range of ecosystems, including peatlands, grasslands, and arable in temperate and tropical regions. She employs various techniques in her research, including X-ray computed tomography, stable isotope labeling, microbial phenotyping, novel organic matter characterizations, and a suite of standard biogeochemical and ecological measurement approaches. So Hannah, I invite you to share your screen, and whenever you're ready, take it away, please.
Thank you, Saoirse, and thanks for inviting me to present at this session. So before I begin, I'd like to acknowledge the collaborators across this body of work, so including the University of Nottingham, where most of this research was undertaken, and the British Geological Survey in the UK, and collaborators in Brazil, such as Embrapa and Sao Paulo State University. So I wanted to start this talk by sharing kind of the wider context of this topic, and then I'll work down to discussions on the research and mechanisms which govern greenhouse gas release from soils. So soils provide a whole host of ecosystem services.
And I'm sorry, I can't see your screen yet. Have you shared your screen?
Yeah.
That's brilliant. I can see it now.
Oh, you can see it now. Cool. We did practice this, everyone, I promise. So I'll just go back to my first slide and just again acknowledge the collaborators. So University of Nottingham, British Geological Survey, and collaborators in Brazil, such as Embrapa and Sao Paulo State University. So I wanted to start this talk by sharing the wider context of this topic, and then work down to discussions on the research and mechanisms which govern greenhouse gas release from soil. So soils provide a whole host of ecosystem services, which you can see on the figure to the right from the FAO. So some services include food production, water purification, and moisture retention. But today I'm going to focus on these three services and show how soil in the right management and climate can provide a means of carbon sequestration, climate regulation, and nutrient cycling. So how important are soils in greenhouse gas production? So this figure is taken from Climate Watch and demonstrates the impact which agriculture, forestry, and land use has on global greenhouse gas emissions. Specifically, agricultural soils contribute to at least 4% of total greenhouse gas emissions. And some of the work I'm going to show you today provides some evidence and mechanisms in which, under the right soil type, management, and climate, agricultural soils can work towards mitigating climate change rather than exacerbating. So the management which I'm going to focus on today is no tillage or zero tillage. So this is the practice by which the farmer or landowner will pass over the field maybe once or twice, causing minimal disturbance to the soil, leaving at least 30% of the previous crop residue on the surface, and direct drilling into the previous stubble. So this cultivation technique has many benefits to the soil in reducing erosion through wind and rain, and reducing the fuel and labour costs due to reduced number of passes across the field. However, there is a lot of contradictory research in the literature surrounding, firstly, carbon sequestration. So does zero tillage actually sequester more carbon, or is it just concentrated in the surface? Nitrous oxide emissions. So zero tillage might reduce carbon dioxide emissions from the soil, but at the same time, due to the higher moisture content, it might increase nitrous oxide emissions. And this is a greenhouse gas 298 times more potent than carbon dioxide. So when you're looking at the balance, how does this impact your kind of global budget from no-till systems? And crop yield. So when does no-till yield more? So there's a lot of contradictory literature surrounding if you might get a penalty in crop yield if you undertake no-tillage compared to your conventional systems such as ploughing. So now I'm going to talk you through some research which I undertook to try and provide some evidence to these research gaps. So this is a map showing the percentage of arable land under no tillage globally. And as you can see, the cultivation technique is most commonly practiced across North and South America. So the first research that was carried out was in the East Midlands in the UK, where only about 7 to 11% of land is under no tillage. So firstly, I went to 32 farms and sampled a total of 160 fields. So it was a paired design where I had a no-tilled field right next door to a conventionally tilled field. So I had 80 of these pairs. So theoretically, everything was the same in terms of soil texture, cropping system where possible, microclimate. The only difference was the management of which this soil was under. And what was really important to note here was because I had so many fields of no-till, this incorporated different lengths of duration that the soil had been no-till for. So some of the soils had been in zero tillage for 1 year and they were recently converted, whereas other soils had been in zero tillage for up to 15 years. So we took a suite of different soil samples for analysis. So we took intact soil cores, so they were 5 centimetres in diameter, 30 centimetres deep. We took these intact soil cores and we incubated them to get greenhouse gas emissions at different temperatures. We scanned them using X-ray computed tomography. We measured carbon and nutrients down to 50 centimetres and a whole host of other analysis as well. So these are an example of some of the soil cores that we took across the East Midlands. And even with the naked eye, you can see that there are visually differences between these two soil managements. So an example on the left, you've got a no-tilled, and on the right, you've got a ploughed soil. And again, here from a bird's eye view, you can see the differences. So these soil cores are taken maybe 5 meters apart, and it's the management that this system is under that is changing these soils. So we can use a technique called X-ray computed tomography, and it's now commonly used in soil and plant science to be able to non-destructively visualize soil and soil pore architecture or root development. And crucially, this provides us with a mechanism to potentially be able to relate soil structure from maybe 1 micron up to 60 or 100 microns to ecosystem services such as carbon storage and greenhouse gas emissions. And then hopefully this loads. So this is an example of soil cores subject to X-ray computed tomography. And what you can see here in the red is the soil pore architecture. And you can see the huge differences between these soil columns. So on the left, you've got the zero-tilled soil column. And you can see here a really highly connected system. Whereas on the right, the ploughed soil is a lot more porous, but not necessarily as connected. Again, this is showing some more images of this connected porosity and the differences between the two systems. So if we have a look at the graph on the right, These are the soils taken from the conventionally tilled, the ploughed systems. And you can have a look at the detectable porosity, which is in yellow, and the surface connected porosity. So say you have a soil column this length, the surface connected porosity is if you have a pore up here and a pore down here. Is this connected in some way through your soil profile? So you can see under the conventionally tilled systems, you have a high porosity overall, but a lot of them aren't necessarily connected to the surface. And this is going to have implications for your greenhouse gas emissions, your water infiltration, and just your general transport through your system. After 1 to 5 years in no tillage, things seem to actually get a little bit worse in terms of surface consolidation. Your detectable porosity is now decreased in line with your surface connected porosity. But give the system some time to regenerate that structure. If you're not constantly passing over the field with a plough, things are left in place to develop, such as your root systems and your biopores generated by your earthworms. And this can start to regenerate your soil structure and starts to create this surface connected porosity. And then after 11 to 15 years, you can see the system improves further in that your overall porosity and the surface connected porosity are now increasing. So what was quite unique about this experiment is that we could directly relate and use the same soil cores, which we'd measured the structure using X-ray CT, and incubate these soil cores under a range of different temperatures to have a look at their greenhouse gas emission production. So in the blue, you've got the conventionally tilled, and in the green, you've got your time series on years under zero tillage management. And so for carbon dioxide, you can see there's a huge pulse of carbon dioxide from the conventionally tilled soils. And then after a series of 11 to 15 years in zero tilled systems, you can see that the carbon dioxide is severely reduced from this management system. And then if we look at nitrous oxide, so this is the more potent greenhouse gas, you can see that this conforms true to literature in that the N2O production tends to be a lot higher under no-till systems because you've got the crop residue left on the surface, which is a perfect substrate, and you've got a higher moisture retention, which again is a perfect environment for N2O release. And then what we did was use this formula to calculate something called global warming potential, which takes into account the potency and the amount of nitrous oxide emissions, carbon dioxide emissions, and methane to generate your global warming potential. And what you can see here is that when we're looking at the paired design, so after 1 to 5 years, there's no significant difference in the global warming potential between the two systems. But then after 6 to 10 years, you see a slight decrease in the global warming potential from your zero-tilled fields. And then after 11 to 15 years, there is this huge significant difference in the global warming potential between these two management systems. And then we also wanted to have a look at total carbon. So if carbon dioxide emissions are decreasing from zero-tilled fields, what's actually happening to the carbon storage within this system? And when looking at the fields as a whole, so comparing 80 no-tilled fields against 80 conventionally tilled fields, there was no significant difference in carbon stored within your soil profile. But if we separated this out again into time, so duration in no-tillage, you can see this temporal effect again through your systems. So you can see after 11 to 15 years, there was a significantly higher proportion of carbon in the no-till system compared to the conventionally tilled system. And where is this accumulation happening? So after 6 to 10 years, there was a larger proportion of carbon in the surface, so in the top 0 to 10 centimetres. And then after 11 to 15 years, again, you're seeing this increase in carbon accumulation in the 0 to 10 centimetres. But then you're also seeing it now deeper in the soil profile. And this heavily linked to the structural properties that I showed you earlier. So with these highly connected pores, the organic material can move down through your soil profile and begin to accumulate further down. So that was kind of a whistle-stop tour of the study undertaken in the East Midlands. And then I was very fortunate enough to be able to go out to Brazil as well, where no tillage has been practiced for a lot longer on a wider scale across Brazil. So I went to Maranhão in the north, where they've been practicing zero tillage for 15 years. Goiânia, so I'm visiting Embrapa, where they've been zero tilling for 22 years. Botucatu, so Sao Paulo State University for 30 years, and Ponta Grossa, 28 years. And we kind of did a similar sampling regime, taking these samples, CT scanning them and measuring the greenhouse gas emissions and carbon storage as well. So I'm not going to go too much into this because we don't have enough time, but again, a very similar story is shown across these soils, and it's the additive effect of zero tillage on soil properties. So the graph on the left, you can see again this translation of carbon moving down into your soil profile after 31 years. And then on the right, you can see the carbon dioxide emissions with time under no tillage, and you can see a really nice decrease with time under no tillage. So something else I wanted to explore was, well, what's happening with this carbon? So you're seeing a greater carbon accumulation in long-term no-till, but a lower decrease in CO2 emissions as well. So what is happening to this carbon? So we sieved out, once the soil cores were scanned and greenhouse gas emissions done on them, we sieved out and took different aggregate size ranges. So in this example, it's macro aggregates. And we had a look at the kind of the finer scale porosity and architecture of this soil. And the yellow here is the pore space within these macro aggregates, and that's under conventionally tilled soils. So these aggregates tended to have a higher total porosity. And then what you can see here is kind of their paired examples, their paired counterparts. So you've got 2 years in no tillage, 15 years in no tillage, and 31 years in no tillage. And you can see a stark decrease in the kind of internal porosity within these aggregates, which may act as a kind of a physical mechanism for this carbon to be stored within these systems. We then use something called a Rockeval pyrolysis to have a look at the total organic carbon and the thermostability of this carbon within these soils. So any kind of perturbation that these soils are exposed to, what happens to that carbon? Is it immediately released into the atmosphere, or is it held on a bit tighter into the system and not released as carbon dioxide? So this graph is showing total organic carbon, and you can see the greatest increase in total organic carbon happened in the 30 years of zero-till soil. And then if we have a look at this, the Rockeval data looking at the thermostability within these macro aggregates, you can see that the proportion of recalcitrant carbon increases by 1.7% in soils that have been in tillage for 15 years. And again, this proportion of recalcitrant carbon, the carbon that's going to stay in your soil for longer, increased by 3.2% in soils that have been in zero tillage for 31 years. Now, these might seem like quite small numbers to be increasing by, but across the data set, it was significantly larger compared to the conventional tilled counterparts. So I was trying to do kind of a graphical abstract or a summary of what the data shows, comparing conventional and zero-tilled soils. So unfortunately, within the study, we can assess crop yield across all of the farms, but it's something we're working on. You can see not only does the quantity, but also the quality of carbon changes under long-term no tillage. You get a reduction in your global warming potential under long-term no tillage, So yes, nitrous oxide emissions increases under long-term no tillage, but this doesn't offset the benefits that no-till can provide. And in terms of soil structure, you get a greater surface connected porosity through your soil profile, which could potentially be a mechanism for how the carbon and organic matter can move deeper down through your soil profile and accumulate. So with that, I'll say thank you very much.
Thank you very much, Hannah. That was excellent. I think it's going to generate a lot of questions, so we'll hold those questions to the end. And if anyone has any further questions for Hannah, please put them in the Q&A. Okay, thank you. So our next speaker is Dr. Paul Murphy. So Dr. Paul Murphy is Associate Professor in Soil Science in the UCD School of Agriculture and Food Science and a member of the UCD Earth Institute in UCD. His research interests are in soil nutrient cycling and management relating to soil management practices, water quality and greenhouse gas emissions, with a particular interest in aspects of spatial and temporal variability and scale. His recent work has included the use of N-isotopes and isotopomers to investigate sources of soil N2O emissions. Paul has over 20 years experience in research and teaching in this field and has published over 40 peer-reviewed scientific papers. So with that, thank you, Paul. I invite you to share your screen and then take it away whenever you're ready.
Okay, thanks, Saoirse. And please let me know if my PowerPoint slides don't show up. I'll try and set that up now.
Yeah, that's good.
Okay, great. Thanks. So again, and thank you also for the opportunity to speak today. So I'm going to continue on that theme of soil and greenhouse gas emissions and focus a bit more on nitrous oxide emissions and some of the work that we've been doing with a range of colleagues in UCD and elsewhere in this area. Okay, similar to Hannah, I'm going to start out with a global picture and then zoom into the soil, but my research more or less focuses on soil and the Earth's surface system and the critical role that soils play in that. So just to set that bigger scene, if you like, soils, or the pedosphere, is just a thin skin on the surface of the terrestrial Earth, but it plays a key role in some of the most important biogeochemical cycles, because soil is the space and time where the other spheres in the Earth's surface system overlap. So the atmosphere, the biosphere, the lithosphere, and the hydrosphere. Soils then regulate fluxes of mass and energy between these other spheres. And how we manage soils, for example, as we've just seen with cultivation practices, or as we'll talk about now with fertilizer application to soils, affects those fluxes. And of course, for most of the history of agriculture, we have focused on the link to the biosphere, and most specifically the crops that we are interested in growing. But of course, we've become more aware of the impacts of how we manage soils on these other parts of the Earth's surface system, as we look at now, in, for example, how nitrogen fertilization can affect nitrous oxide emissions. So as already mentioned, nitrous oxide is a very potent greenhouse gas and is emitted from soils mainly through our management of nitrogen. In fact, the human disturbance of the global nitrogen biogeochemical cycle is such that industrial fertilization or industrial fixation of nitrogen into nitrogen fertilizers, which are then applied to our soils, now roughly matches, by some estimates, the total biological nitrogen fixation in soils at a global scale. So that's a huge anthropogenic disturbance to one of the most important biogeochemical cycles. And that has widespread implications, including emission of N2O, potent greenhouse gas. In fact, that disturbance to the global nitrogen cycle is part of the Anthropocene and the great acceleration that some of you may be familiar with post-1950, and this data would show that here. So post-1950, you can see in the upper figure, for example, a large increase in nitrate concentrations in Greenland ice, and a large shift also in the isotopic composition of that nitrogen. And this is thought to be related to the increased use of nitrogen fertilizer globally, and also processes of nitrification as a source of N2O emissions. Focusing in a little bit more locally, the greenhouse gas emission profile of Ireland is somewhat unusual for a developed country in the proportion that is made up of from emissions from agriculture. So in 2019, 35%, and that is as a proportion increasing year to year. So, and of that, those emissions from agriculture, roughly a third of that is associated with N2O emissions associated with our nitrogen management. So why nitrogen and N2O? Why is that important? Because of what I just mentioned there, but also this study here from Teagasc, a marginal basement cost curve analysis of different mitigation options for greenhouse gas emissions from agriculture. Just very briefly to explain, if you look along the bottom, you have cumulative abatement of CO2 or greenhouse gas emissions, and up the side you have costs associated with that emission or abatement. And you can see for a number, if I highlight just the fertilizer type, clover, and nitrogen use efficiency are all related to our nitrogen management, and they have a relatively large potential abatement of greenhouse gas emissions and are close to cost neutral, which would be about the zero line here, or indeed potentially cost beneficial to the farmer, which would be below the line. So some of these mitigation measures, just to highlight from research that I've been involved in, looking at reducing surplus nitrogen use efficiency. In the Agri-Benchmark project, an EPA funded project, we looked at using National Farm Survey, so nationally representative data from different farm systems in Ireland to assess their contribution to the surplus nitrogen. So surplus nitrogen at a farm level being an indicator of nitrogen source pressure would tend to be associated then with greater, higher nitrogen surplus associated with greater potential for nitrogen loss to the environment, including, of course, as N2O emission. And you can see the importance of dairy as contributing to that surplus. So 43% of the estimated national nitrogen surplus was associated with dairy, and that's on 21% of the land area. So if we were to sort of choose where we need to focus to really improve our nitrogen management and reduce our surpluses in order to reduce the losses to the environment, dairy would be a good place to focus on. Okay, also from that work on using National Farm Survey data, this looked at dairy farms in particular, and it's the farm gate nitrogen surplus. And along the bottom there, you can see milk production in litres per hectare. So increasing milk production per hectare as you go to the right, and up the left, you can see nitrogen surplus in kilograms nitrogen per hectare. So increased nitrogen source pressure as you go up on the left. As you might expect, as milk production increases per hectare, so does nitrogen surplus. But what I find more interesting about this data is the spread that we can observe in these farms. So for example, two farms both producing at the same level of output per hectare, 10,000 litres per hectare. One doing that at a nitrogen surplus of around 50 kilograms of nitrogen per hectare, and the other closer to 300 kilograms of nitrogen per hectare. That's a very large difference in the nitrogen source pressure, and likely a large difference in resultant losses of nitrogen to the wider environment, including as N2O emissions. Lots of factors, of course, would affect this, but one of the important ones would be management. And one of the areas or aspects of management that we can look at potentially to reduce that surplus nitrogen, reduce fertilizer nitrogen use, make it more efficient, is the use of grass, clover, and multi-species swards. So for the rest of the results I present here will be from related to the Smart Grass project, which was headed up by Helen Sheridan, and I only really kind of jumped on the bandwagon when I joined UCD. But this looked at plots, which you can see there, a whole range of sward compositions from perennial ryegrass monoculture to different combinations of grass, clover, and herbs in different proportions. Just to focus in on the one result from that over the 3 years of the experiment, when we looked at the estimated N2O emissions, which you can see up the left-hand side there in kilograms of N2O per ton of dry matter per hectare per year. There's a large difference in the estimated N2O emissions from the perennial ryegrass monoculture, the sort of conventionally managed at 250 kilograms nitrogen, fertilizer nitrogen that you can see here, compared to the ryegrass clover at 90 kilograms of fertilizer nitrogen, the middle bar, or a simple or more complex multi-species mix also at 90 kilograms of fertilizer nitrogen per hectare per year. In fact, the amount of N2O emitted per kilogram of or per ton of dry matter herbage produced was about 9 times higher for the perennial ryegrass at 250 kilograms of fertilizer nitrogen compared to the grass clover and the multi-species. So very significant potential savings in N2O. Why that is because of a number of factors. One, the grass clover and multi-species swards were able to roughly match the perennial ryegrass at 250 in terms of the overall dry matter yield, but obviously at a much lower fertilizer nitrogen level input. And that fertilizer nitrogen was applied early in the year and in the form of urea, whereas fertilizer nitrogen applied to the perennial ryegrass sward was applied throughout the year, as would conventionally be done, and as calcium ammonium nitrate in the later months of the year through the summer and into the autumn. So to calculate these estimated emissions, we used an emission factor derived from the literature, in large part from the work of Mary Harty, who looked at N2O emissions from different fertilizer forms. And we assumed then an emission factor for CAN of 1.5% and a much lower emission factor for urea applied in the springtime of 0.25%. So those different factors account for this large difference in estimated N2O emission, direct N2O emission from fertilizer nitrogen use. Okay, so the rest of what I will present here is largely from the PhD work of Conor Bracken, a PhD student who worked with us on that project, and looked then at those plots, or a selection of those plots, using the static chamber method, which you can see there, taking the gas samples from the headspace and then GC analysis to determine the N2O concentration and emissions, but also used a more novel cavity ring down spectroscopy, the Picaro instrument you can see here on the right, which looks at both the N2O concentration in the gas samples, but also the isotope composition and also the isotopomer composition of that N2O. And this allows to lift the lid a little bit on the processes that might be leading to those N2O emissions, as we'll see. Okay, soils are very important, hopefully a message that will come through from these two presentations, and all of this was carried out on one soil type, so that's an important caveat to bear in mind. This is the soil at the UCD Lyons long-term research farm, a clay cluvisol. You can see the mottling in the lower part of the soil profile there. Clay loam, quite heavy textured, has fairly restricted drainage. That's a key component of it. Also, it's about pH neutral due to the limestone parent material. These plots are part of a mixture experiment. It's illustrated here. Mixture experiments are different to what you might be used to in terms of replicated and blocked plot experiments. A mixture experiment establishes mixtures of, in this case, sward composition, and allows you then to analyze both the main effect or individual effect of, in this case, species, which would be perennial ryegrass, white clover, or plantain, but also any diversity effects which might emerge from interactions or complementarity between those different species mixes within a sward. And so in this case, the end points of this triangle was grass, 100% perennial ryegrass, a grass clover mix, white clover of 40% grass, 60% clover, and a grass clover herb mix, a grass and herb plantain mix of 40% grass, 60% herb, and then you see the other compositions intermediate in between. And in this case, looking at the effect of these on N2O emissions following urea application. So Conor applied urea to these plots during the summer and then monitored the N2O emissions for the following 2 months. And we were interested to see what the effect of soil moisture conditions, again, chiming with what Hannah was just talking about there. We know that N2O emissions from soil tend to be very sensitive to soil moisture content, soil moisture conditions. So Conor manipulated the soil moisture conditions in a split plot. So the one half of the plot was under ambient conditions or dry conditions, and the other plot was wetted to get it up above 60% water-filled pore space. 2 things to take from this figure. If you look along at the right-hand side, and the overall N2O emissions in grams of N2ON per hectare, you'll see that emissions tend to be higher under the wetter soil conditions. So as expected, wetter soil conditions tending towards greater denitrification in the soil and encouraging greater N2O emissions than under dry conditions on the left-hand side. The other thing to note is that as you go towards the legume corner, the bottom left-hand corner of the triangle, in both cases, you get towards higher N2O emissions. So higher legume content associated with higher N2O emissions. Remember that this is urea being applied to these plots during the summer months when biological nitrogen fixation is likely high, and the possible interpretation of this would be that with higher legume content in the sward, fixing more nitrogen, and then that nitrogen being mineralized and released, you have more mineral nitrogen available in the soil solution in those plots. And then when you're applying urea fertilizer on top of that, if you like, supplying more mineral nitrogen, more of that is then vulnerable to being emitted as N2O rather than taking up into the crop. So the importance of appropriate fertilizer nitrogen management to the sward composition, especially the legume content of the sward. I mentioned that Conor was looking at the isotopes and isotopomers. So just very briefly, we don't have time to go into much detail, but you can have 14, 15 N isotopes of nitrogen, but within the N2O molecule, that nitrogen can be in either the alpha or the beta position within the N2O molecule, and both the isotope and the isotopomer measurements can be an indication of what process actually led to the N2O emission. So just to draw your attention to the key points from these 2 figures, if you look on the left-hand side, which is under ambient or dry conditions, there is a significant effect as you go to the herb corner of the triangle there, the plantain corner of the triangle, with higher site preference indicating potential biological nitrification inhibition. This is something that is hinted at in other lines of evidence in the literature, that plantain may have an inhibitory effect. There are some chemicals that plantain contains that may inhibit biological nitrification and therefore slowing down the release of nitrate and slowing down in turn N2O emission and also potentially nitrate leaching. So it's an area of real interest. and some evidence there for that in those multi-species swards containing plantain. And on the right-hand side, under wet conditions, a strong trend towards, with increasing legume content, decreasing site preference value, indicating potentially greater contribution of denitrification to N2O emitted. Looking across those plots in that study, in fact, and using the isotopes and the isotopomer data to fractionate the N2O emitted into sources, whether nitrification or denitrification, and then looking at the relationship to water-filled pore space, estimated water-filled pore space in those plots. There appeared to be a kind of a threshold value around 65% water-filled pore space in which denitrification becomes dominant over nitrification. And that actually is in agreement with other studies from the literature. Something like 60 to 70% water-filled pore space seems to be something of a threshold beyond which denitrification becomes more dominant as the source of N2O. And that is typically associated with greater N2O emissions, importantly. Okay. Conor then went on to look at this in a lysimeter study in the Environment Research Centre in Rosemount here in UCD, where he took those soils in small lysimeters, as you can see here, and was sampling again over a whole year, this time the greenhouse gas, the N2O emissions from these different swards. And just very briefly, because I'm conscious of time here, the cumulative N2O emissions from those swards, you can see in this graph. So perennial ryegrass and low white clover, perennial ryegrass and high white clover, and then a simple multi-species mix of ryegrass, white clover and plantain, managed at different fertilizer nitrogen levels appropriate to the clover content of the sward. You can see the fertilizer nitrogen levels at the top there. What he found, in fact, was no significant difference in the cumulative N2O emission. You'll notice on the right-hand side here for the simple multi-species mix at 45 kilograms of fertilizer nitrogen is numerically lower than the others and was borderline significant, almost significantly different, but not in fact. So what I'll show you in the last figure after this is that, in fact, at a key time of the year when most of the nitrogen N2O emissions happened, the emissions were actually significantly lower from that treatment. But first of all, to draw a conclusion from this, why would that be, given the different levels in fertilizer nitrogen? Well, we chose those levels of fertilizer nitrogen application to try to match, if you like, the clover content of the swards to maintain a similar level of dry matter yield. And so although the fertilizer nitrogen input is much lower on the grass clover and multi-species swards, they of course have a large amount of nitrogen being fixed biologically due to the clover content, and at least some of this clover, some of this nitrogen fixed biologically is apparently vulnerable to N2O emission after mineralization. Okay, and that would be in agreement with other studies in the literature which also have found higher N2O emission associated with greater levels of biological nitrogen fertilization at higher clover content in the sward. So the balance of these things is important, balancing fertilizer nitrogen to the clover content and trying to minimize that fertilizer nitrogen input as much as possible. Okay, so very briefly, over time, over the year or so of that experiment, what this figure shows here is daily N2O fluxes along the bottom, grams of N2O per hectare per day. And highlighted there at the arrows is when fertilizer nitrogen was applied. And what you can see is that N2O emission, first of all, is quite temporally variable. This is what is generally observed. It's highly temporally and also spatially variable, which makes it a bit tricky. but peaks are clearly associated with fertilizer nitrogen application and also with wet soil conditions. So N2O emissions tend to be driven by fertilizer nitrogen application and also by wet soil conditions. So you can see that the peaks following the application of fertilizer nitrogen, often with a bit of a time lag. Again, that's quite commonly observed. The peak of emission may often come a bit of a time later than the fertilizer nitrogen application when the soil is wetted by rainfall. And you can see in the figures above the rainfall input and the estimated water-filled pore space for these soils at different depths. The other thing to note is on the left, where I've highlighted in the sort of brown color, conditions in the soil were much drier. If you look at the water-filled pore space above, much drier. In fact, that was the summer of 2018, which some may recall was a fodder crisis due to an extended period of drought. So it's quite actually unusual conditions. That was then followed by a wetting up cycle through the autumn and into the winter, and water-filled pore space, soil moisture levels remained higher then through the rest of the next year. And you can see how that was generally associated with higher N2O emission. And also in that period, in the spring of the following year, the difference between the different sward types became much more pronounced under those conditions. In fact, in that key period of high N2O emissions in the spring, the perennial ryegrass and high white clover and the multi-species swards had significantly lower N2O emission than the perennial ryegrass, around 40% lower. So very significant lower N2O emissions. So the take-home message from that is the importance of the antecedent soil moisture conditions, the rain that will follow following fertilizer application, and the timing of nitrogen fertilizer application in determining N2O emissions. Okay, I'm very conscious of time, so I'm just going to click to some key take-home messages. So nitrogen management can provide a route to mitigate greenhouse gas emissions that can be quite direct. Any management practices we can find and implement that can reduce, in particular, our fertilizer nitrogen management can be quite easily incorporated through appropriate activity data and emission factors into the National Greenhouse Gas Inventory. Wet soil conditions, more than 60 or 70% water-filled pore space tend to drive higher N2O emissions associated with denitrification. The availability of mineral nitrogen, whether from fertilizer nitrogen applied or from organic matter mineralization, release of nitrogen from biologically fixed nitrogen is also key. N2O may be emitted via nitrification and denitrification concurrently in the same soil. Don't go into it here, but perhaps in different range of pore sizes, sort of chiming with what Hannah was talking about there about the detailed structure of the soil, the porosity in the soil, connectivity of pores may be important, and at different levels in the soil profile. So it's entirely possible that you could have nitrification and denitrification happening in different parts of the porosity of the soil and at different levels in the soil concurrently. It's not just about surface soil moisture conditions, 0 to 10 centimeters. We tend to focus a lot on that surface soil with very good reasons as well. But conditions deeper in the soil are also very important. As a general observation from that experiment, we should avoid high fertilizer nitrogen applications during the spring, particularly if the soils are wet. And for the management of grass, clover and multi-species swards, as we saw there, the fact that they have lower fertilizer nitrogen application, significantly lower fertilizer nitrogen application at that time of year, may be a particular advantage in terms of reducing N2O emissions. Okay, thanks very much for your attention, and I'd be delighted to take any questions that there might be there as well.
Thanks for that, Paul. And yeah, just a reminder, any further last questions, please put in the Q&A. So perhaps, Paul, yeah. So first off, welcome back, Hannah, as well. We just have a couple of questions. I'll start with Hannah first. could you tell us what instrument or technique you used to measure greenhouse gas emissions in the zero-tillage experiment?
Yeah, sure. So it was the same soil cores that we took from the fields, and then we just incubated them in, it's very technical, but it looked like long containers that you store spaghetti in, to be able to put the cores in there. And then we had a super seal on top and made sure the lid was all sealed round. And then we had a magnetic stirrer at the bottom because carbon dioxide tends to sink. And then we took a sample from the top at 0, 5, 10, 20 minutes using a syringe, and stored them in exutainers 12 ml, and then analysed them using the GC.
Okay, thank you. So another question for you, Hannah. You showed the carbon concentration increased with time after conversion to no tillage,
Yep.
But what about the stock, i.e. accounting for changes in bulk density?
Yeah, exactly. So that's where a lot of the confusion comes up in the literature. So we took this into account and didn't necessarily calculate the stocks using bulk density, but the equivalent soil mass method. So there was a paper in EJSS from, I'm going to say the name's wrong, but Horton Wesner, and they provide an Excel spreadsheet to be able to take these changes in bulk density between the two systems into account. Yeah.
Okay. And could you comment on the methane uptake results?
Yeah, so that's something that's actually quite interesting from this study, and something I want to look into further. So within this study, there were no significant differences in methane uptake, but there was an interesting, like a visual trend that you could see that methane uptake was increasing with time. So there is some literature that says that methanotrophic activity in agricultural soils can kind of repair itself in agricultural systems, but that can take up to decades for any kind of effect to be shown. So yeah, that's something I'm interested in, maybe looking at even longer term no-till soils to see if we can see something going on there. Yeah.
Thank you. And now a question for Paul about the clover N2O emissions. The question asker has asked, could this simply be because of the sparing use of soil N, given most N may be obtained through N fixation, so that more is available for conversion to N2O?
Can you say that again, or can I read that somewhere? I didn't exactly follow that.
In the Q&A.
Okay.
The last question.
Yeah. I wasn't really clear about the clover N2O emission story. Okay. Could this be simply because of the sparing use of soil nitrogen, given most nitrogen may be obtained through nitrogen fixation? Yes. So that more is available for conversion to N2O. Yeah, I think so. As in the higher the clover content of the swards, the more biological nitrogen fixation there is. And the more over time, especially as that clover is active in the soil over a number of years, there will be a sort of build-up of the soil's ability to mineralize and release nitrogen into the soil solution. So then if you are applying fertilizer nitrogen, that nitrogen then is not likely to be, well, depending on the balance of these things, but you could think of it that the sward basically doesn't need that nitrogen. It's already acquiring its requirement of mineral nitrogen for uptake from the soil itself, and then more of that nitrogen is not taken up by the plants and then is vulnerable to being lost by different pathways, including N2O. So that would be why the management of nitrogen that we used in those experiments, and that will be advised as well, would be that if you're applying fertilizer nitrogen to grass clover or multi-species swards, that you apply earlier in the year when biological nitrogen fixation is when clover is much less active, and that can support the growth of, for example, the perennial ryegrass. But then we would have applied no fertilizer nitrogen past May, and from that point on, the nitrogen requirement is met by the mineral nitrogen present in the soil. I hope that answers that question.
Thank you. I just have a question for Hannah, but maybe Paul can come in as well. So Hannah, you mentioned that the nitrous oxide emissions were greater from the zero-tillage plots. Did you find typically that the zero-tillage plots were wetter? So would that link back to some of Paul's findings?
Yeah, definitely, exactly that. So across all of the fields, regardless of years in zero-tillage, because you've got that crop residue left on the surface, and that really retains the water and increase the soil moisture. So definitely, yeah.
So we have maybe time for just one quick final question. So this is to Hannah again. So can we say that there is no immediate 0 to 10 years benefits in terms of carbon sequestration? It seems like significant differences were observed in the long-term zero-till field of 31 years.
Yeah, so this is definitely the trend that was coming across in these study sites. So it is really important to put a caveat on this, in that the literature regarding no-till is so contradictory, and it's because it is so dependent on loads of factors: climate, texture, previous cropping history, cropping rotations, so many different things. But from this study site across the 160 fields, that is the trend that we can see. So we could after 6 years see an increase in carbon sequestration, more predominantly in clay textured soils. It took a lot longer for the response to be seen in sandy textured soil, but it became like statistically significant after 10 years. Yeah.
Okay. Thank you very much. So with that, I'd just like to thank both the invited speakers for excellent talks today. Thank you to all the participants that came along today. And just a quick reminder that this recorded webinar will be uploaded to the UCD School of Agriculture website in the coming weeks, if you want to watch back. So thank you everyone again for your time, and take care. Bye-bye.